Hello, everybody. Today we are going back to our exploration of the different types of organic molecules. We've spent almost the last week and a half on proteins because they're one of the biggest, most diverse groups, and they're really important, especially those enzymatic proteins that we were focusing on last week. But this week we're going to move on and take a look at the carbohydrates, the lipids, and the nucleic acids. So we're going to start today by going back to your pattern matching activity. Hopefully you still have your molecule cards. And today you're going to start by sorting out the ones that are examples of sugars. So take a few minutes and go and work on that and come back to this when you have finished. All right, keep your molecules nearby as you go through the rest of this little lecture video today, so that you can look at those examples you have and kind of notice the things that we're talking about on the molecule examples that you have. So sugars are classified into different groups depending on the complexity of the molecule. And the words we use for sugars often contain this word saccharide. Saccharide just basically means sugar. So the building blocks, the monomers that make up carbohydrates, are called monosaccharides, which just simply refers to one sugar molecule. So most monosaccharides, which are not all, but most of the ones that you have in your molecule kit, are multiples of the formula CH2O. In other words, they have equal amounts of carbon and oxygen atoms, and they have twice as much hydrogen. So if you think about things like glucose, C6H12O6, that might be a monosaccharide that you remember the formula for. It follows that pattern. It has 6 each of carbon and oxygen, and twice as much, it's got 12 hydrogen. So this isn't a specific monosaccharide, but it's kind of the generic formula for monosaccharides. Most monosaccharides and sugars in general, carbohydrates in general, have a carbonyl group. Remember, that's carbon double bonded to an oxygen, and multiple hydroxyl groups attached to the carbon skeleton. So as you look at your examples in your molecule set, see if you can recognize those features. Those are some things to look for to help you recognize carbohydrates. In terms of the molecules themselves, carbohydrates are often have names that end in OSE: glucose, fructose, lactose. Those are all different types of monosaccharides. Again, it's good to just notice those name endings because that can help you kind of figure out what type of molecule you're talking about if you ever see it in an FRQ or multiple choice question. Monosaccharides can be in a linear shape or they can be in a ring shape. And you have some of each of those within your molecule collection. In aqueous solution, so that means when dissolved in water, the ring structure tends to be a little more stable, which is why you usually see glucose, for example, drawn as a ring when we're talking about it as a fuel for living things, because that's how it exists most of the time. Glucose, of course, is the one we talk about a lot in biology because it's the main energy source for cellular respiration. So we will get back to that one when we talk more about energy later in the class. If you link two monosaccharides together, you get what's called a disaccharide. The prefix di means two, so it's two of those monosaccharide monomers joined together. And just like with our proteins a long time ago, these monomers are also joined by those dehydration reactions. So you take those two sugar monomers, you remove a hydroxide from one, a hydrogen from the other. So you take out a water molecule, which bonds the two carbohydrate monomers together. In carbohydrates, because you're linking different monomers, there are different atoms and different functional groups that actually form that bond. So the bond itself gets a different name. In carbohydrates, that bond formed through a dehydration reaction is called a glycosidic linkage. And I kind of remember this one because glycosidic sounds kind of similar to glucose, which is a carbohydrate that a lot of people know. So a glycosidic linkage is the bond that's formed between two carbohydrate monomers. Examples of disaccharides are sucrose, which is just basic table sugar, and lactose, which you may have heard of. It's a sugar in a lot of dairy products that some people can't break down. Those are people who are lactose intolerant. And so here is an example of kind of how sucrose is formed. Sucrose, that's regular old table sugar, is glucose, that's one of the monomers, and fructose. Each of those, glucose and fructose, are monosaccharides. They're just single ring sugars. And when you join them together, this bond through the middle is that glycosidic linkage formed by a dehydration reaction. You form this disaccharide that is sucrose. If you join many of these together, then you get a polysaccharide. So these are your complex carbohydrates, those bigger carbohydrate molecules formed of big, long chains of monosaccharides. There are different types of polysaccharides. Some of them mainly function as storage polysaccharides. And when we say storage, we really mean they're storing energy in general. So starch is a storage polysaccharide that's made by plants. It generally has this helical kind of spiral shape. Animal enzymes, so like we, can hydrolyze this polysaccharide. So we do have enzymes that can add the water back in. Remember, hydrolysis is the opposite of that dehydration reaction. We can add the water back in and break down starch into those monosaccharides, into glucose molecules, and use this for energy. So that's why foods like pasta and potatoes and bread that contain a lot of starch are good sources of long-term energy. Another type of storage polysaccharide is called glycogen. That's this kind of branched polysaccharide that you can see down behind my picture. This is made by animals. So this is a polysaccharide that your body builds when you have extra glucose. If you don't use all of the energy that you get from your food, your body stores some of it by building this molecule of glycogen, and you store it in liver and muscles. So this is kind of a backup place to store energy for a middling amount of time. So the glycogen that you have in your liver and muscles is enough to last you and provide you with energy for about a day without eating any food. Beyond that, your body would have to dip into your reserves of lipids and fat molecules and get energy from other sources. There are also structural polysaccharides. So those last two were storage polysaccharides, starches and glycogen. Structural polysaccharides are mainly used as building material. Cellulose is probably the most abundant organic compound on Earth. It is the structural polysaccharide that makes up plant cell walls. It has a really distinct shape. It's just a very, very, very long chain of monosaccharides that gets grouped together into bigger fibers, which makes it really strong. But the structure of this, animals like we are not able to hydrolyze this, to break it down as energy. It's an important part of our diet as fiber. It helps just kind of clear out our digestive system and keep things moving, but we cannot actually break it apart and use it for energy. There are some microorganisms that can digest this, like some that live in the guts of cows. They have particular microorganisms in their stomachs that can break this down. Termites can digest cellulose. That's why they can eat wood, but we cannot. So here you see a picture that kind of shows you the structure, these really long chains that then bunch into these big fibers that make up the cell walls of plants. It's a little kind of an interesting little chemistry tidbit. The only reason we can't digest this is a simple structural change in the glucose monomers that make it up. So the top picture you see here is called alpha glucose. You can see it has this hydroxide on the lower side of the ring. When you link those together, you get these glycosidic linkages that are kind of hanging below the molecule. We do have enzymes that can break this up. This is a starch called amylose that we can break down for energy. But if you simply reverse the position of the hydrogen and hydroxide on this one carbon on this glucose molecule, that changes the shape of those glycosidic linkages. And you can see just a very, very small change in the shape is the only difference between amylose and cellulose. But for whatever reason, we just do not have enzymes that are able to break apart these beta glucose bonds. So that's the only difference, but that shows you how important the shape of molecules are and how specific the enzymes that digest these are. We have enzymes that can digest amylose, but they cannot digest cellulose. So again, just a reminder that that structure-function relationship is really critical, and every little detail of the shape of these molecules matters. Another type of structural polysaccharide that you'll hear about sometimes is one called chitin or chitin. You'll hear it pronounced both ways. Arthropods, like spiders, scorpions, things like that, use it to build their exoskeletons. It's also what makes up the cell wall of funguses, of mushrooms and other types of fungi, molds, mildews, those kinds of things. So that is another type of structural polysaccharide. So that's it for carbohydrates. We're not going to spend as much time. This is a group of molecules we will come back to more when we talk about energy, since they are the major source of energy in living things. That's the main function of carbohydrates. Just a couple of reminders here at the end. So with our pattern matching activity, like I said, we'll be finishing this up this week. So far, you should have separated out all your amino acids and your sugars. We will be finishing the lipids and the nucleic acids later this week. There is a Mastering Biology homework assignment this week. I was a little bit late in posting it, so I made it a little bit shorter than normal, but it is still due Tuesday night. So if you haven't done that, please make sure you go in and do that. And your enzyme lab from last week is due this Friday. Make sure to let me know if you have any questions. Have a great afternoon, and I will see you tomorrow.